Today, we are going all in. We're going to completely unpack the fundamental building blocks of life. We're taking a close look at the science of biochemistry to see exactly how microscopic molecules act as this elegant, incredibly precise assembly line, making everything from your immune system to your bones entirely possible.
Here's how we're going to break it all down today. First, the basics of amino acids. Then, peptide bonds and chains, the amazing functions of proteins, their three-dimensional structure, and finally, protein denaturation and damage. Let's get right into it.
Section 1, Basics of Amino Acids, the individual bricks.
Biology is just incredibly efficient. Out of roughly 300 naturally occurring amino acids out there in the world, our bodies rely on an exclusive VIP club of exactly 20. Just 20! It's wild to think that those 20 basic building blocks are responsible for literally every single protein you need to exist.
Now let's take a look at the actual anatomy of one of these bricks. Notice how that central alpha carbon acts like an anchor? Attached directly to it, you'll always find the exact same three things: an amino group, a carboxyl group, and a single hydrogen atom. But then there's that R side chain. You can kind of think of the R group as the flavor. It's the wild card. Its unique nature completely determines the specific chemistry of each individual amino acid.
And it turns out, nature cares deeply about molecular geometry. Because of how these four groups attach, the molecules can actually exist in two mirrored configurations depending on how they rotate polarized light. You've got the D isomer rotating light to the right, and the L isomer rotating it to the left. If that sounds a bit textbook, just look at your own hands. Your left and right hands are made of the exact same stuff, right? But they're perfect mirror images, and you can't just perfectly stack them on top of each other. It's a brilliant illustration of nature's precise organization, where even a simple mirror image makes a fundamental difference.
Moving on to Section 2, Peptide Bonds and Chains, Linking the Bricks.
Okay, so the crucial point here is scale. As these amino acids link together via what we call peptide bonds, they grow sequentially in size. Snap 2 together, you get a simple dipeptide. 3 makes a tripeptide. Scale it up to between 3 and 10, and now you're looking at an oligopeptide. Between 10 and 50 is a polypeptide. But once you link up more than 50 of these units, boom, you officially cross the threshold into massive macromolecules. These are true proteins that can reach molecular weights well into the millions. To put that into a real-world perspective, let's look at the incredible impact of even the smallest chains. Take aspartame, for instance. It's just a tiny dipeptide, literally just aspartic acid and phenylalanine, yet it packs such a punch that it operates as a potent sweetener in diet sodas. Or look at glutathione. That's a tripeptide, just 3 linked amino acids, but it serves as a massive, vital cellular defender against toxic compounds and physically protects your red blood cells from damage. So yeah, even tiny chains have a huge biological impact.
Which brings us to Section 3, Amazing Functions of Proteins, the machines at work.
Now, rather than just reading off a random laundry list of functions, let's group what these machines actually do. First off, there are internal defenders. Think of antibodies firing up in your immune system or keratin protecting your skin from scrapes. There also are cellular delivery trucks like hemoglobin carrying oxygen or lipid proteins hauling lipids around. Plus, they act as our structural scaffolding and communicators. We're talking bone collagen, cellular receptors, growth hormones. They literally run the entire machinery of your life.
All right, Section 4, Three-Dimensional Structure, Folding into Architecture.
This brings us to a really fascinating pivot point in our explainer. How does a simple, straight string of amino acids magically transform into a highly specific 3D tool? Well, it all comes down to conformation, or structure. That is the ultimate secret to how these proteins actually do their specific jobs.
And it all starts with directionality. Just like reading a sentence from left to right, every polypeptide chain has a strict reading order. It always starts on the left with a free amino group, that's the N-terminal, and it ends on the right with a free carboxyl group, the C-terminal.
Think of this structure kind of like writing a book. Your primary level is the sequence, just the raw letters. The secondary level is how those letters start forming spatial relationships, so your words and sentences. And finally, the tertiary and quaternary levels represent the complete 3D shape. That's the finished, bound book that your body can actually pick up, read, and use.
A perfect example of this in action is the alpha helix, which is a major form of secondary structure. Imagine a tightly coiled, rod-like shape, where those specific R groups we talked about earlier extend outward. And it doesn't just hold this shape by accident. No, it is firmly stabilized by precise hydrogen bonds, connecting exactly the first and fourth amino acids in the chain. In fact, it takes exactly 3.6 amino acid residues to complete one full turn of this microscopic coil, covering a distance of 5.4 nanometers. The absolute mathematical precision of biology here is just mind-blowing.
Finally, let's look at Section 5, Protein Denaturation and Damage, when the machine breaks. To explain this topic completely, we really have to look at what happens when this delicate architecture fails. The formal term for this failure is denaturation. Denaturation is essentially the unraveling of that precise 3D shape, meaning the protein totally loses its secondary, tertiary, and quaternary conformation. But, and this is a really key point, it keeps its basic primary chain intact without breaking the actual peptide bonds. It's kind of like taking an intricate origami swan and unfolding it back into a flat piece of paper. The paper is definitely still there, but the functional shape? Totally gone.
And when exposed to environmental triggers, like excessive heat, for example, the consequences are severe. Because remember, structure equals function. Unfolding means a complete loss of biological activity. Vital proteins like insulin just stop working entirely. On top of that, these once dynamic proteins often become entirely insoluble. They easily precipitate or basically clump out of solution. If you want a real-world visual of this, just think about frying an egg. That clear part rapidly turning into solid white, that is literally heat-triggered protein denaturation happening right before your eyes.
So I'll leave you with this to ponder. If just a simple shift in heat can completely unravel the fundamental building blocks of life, how incredibly delicate is the chemical balance inside your own body right now? It perfectly encapsulates exactly why understanding these microstructures matters so much. From 20 simple amino acids to massive, life-sustaining biological machines, the balance that keeps us alive is precise, it's beautiful, and it's remarkably fragile. Thanks so much for joining me on this explainer. Stay curious and keep exploring the amazing structures inside you.